Hello and welcome back to our videos about the kidneys. In this video, we are going to be talking about reabsorption. Okay, so I just want to start by reminding you that we have four main processes in the kidney. Last time we went over filtration. Now we're going to go over the three remaining processes, reabsorption, secretion, and excretion, one at a time, starting with reabsorption. Okay, so you saw this figure the last time, and what I want to point out on this figure is that these green arrows indicate the process of reabsorption. As you can see, reabsorption happens throughout most of the tubule. However, most of the reabsorption happens in the proximal tubule, okay? You also saw this figure last time. I just want to remind you that most of the fluid that we filter into the tubule gets reabsorbed into the body, right? So just to remind you, we have our blood coming in here through the afferent arteriole, going into the glomerulus, and then leaving through the efferent arteriole. Of the blood that enters the glomerulus, 20% of that is going to filter into Bowman's capsule here. And then we are going to reabsorb almost all of that fluid, okay? You might remember I addressed this question last time. So see if you can remember, why would you want to do this? Why would you want to filter so much fluid and then reabsorb almost all of it back? Think about that for a few seconds. Okay, so the advantage of taking this strategy is that it's an easy way to get rid of toxins and waste materials. Anything that's in your blood that you want to get rid of, it's going to end up in the tubule. Okay, and then what we can do is we can selectively reabsorb back those items that we want to keep in our bodies. Remember that this is destined for the external environment. This is what is going to become your urine. Okay. Okay, so let's take a look at reabsorption from a big picture perspective. Okay, so again, just want to remind you, reabsorption, what we're doing is we're taking materials that are in the tubule, right? They've been filtered into the tubule. We want them to move across the epithelium of the tubule, the wall of the tubule, and back into our extracellular fluid, where it then can re-enter our bloodstream, okay? We're going to be moving molecules in a variety of ways. So transport can be paracellular or transcellular. So just to remind you, paracellular is moving in these spaces between the epithelial cells that make up the wall of the tubule. Okay, transcellular is going to be through, right? So crossing this membrane, going through the cell, and then crossing this membrane here. And our transport is going to be a variety of active and passive transport mechanisms. So hopefully you remember, active transport requires an energy input, passive transport is without an energy input. Okay, so let's take a look at reabsorption again in this big picture fashion, okay? Basically what happens, basically the way that we reabsorb things is we reabsorb sodium by active transport, and then we use that sodium gradient basically to move everything else that we want to move, okay? So this is step one, sodium is reabsorbed by active transport. Next, what's going to happen is well, it's not really, not everything's kind of happening simultaneously, but what is going to happen with our anions is, since we have created an electrical gradient by moving sodium, our anions are going to follow that electrical gradient that was set up, okay? Next, notice that we have moved a bunch of ions from the tubule lumen to the extracellular fluid. What is that going to do to our osmotic gradient? So think about that for a second. Okay, so hopefully you have figured out that we have increased this movement of sodium and anions to the extracellular fluid has increased the osmolarity of the extracellular fluid, Therefore, we end up drawing water out from the tubule lumen to the extracellular fluid, okay? All right, and then finally, now that we are moving water out of the tubule lumen, we're leaving behind a bunch of solutes here. And since we're taking water away, the concentrations of those solutes are increasing in the tubule lumen. Therefore, as a result of that, they follow their concentration gradient out of the tubule lumen to the extracellular fluid, okay? All right, so let's take a look at this in a little bit greater detail. Okay, so taking a closer look at sodium reabsorption in the proximal tubule. Again, what we want to do is we want to move sodium from the tubule lumen across this wall to the interstitial fluid, so to the extracellular fluid. Okay, and we're going to use, overall, we're going to do this by active transport, but certain different steps of it require energy input while others do not. Okay, so, right, so in order for sodium to move across this, from the tubule lumen to the interstitial fluid, we need to cross two membranes, right? We need to cross this membrane right here. This is called the apical membrane, this membrane that's facing the tubule lumen, okay? And then we have this membrane right here, the basolateral membrane, which is facing the interstitial fluid, okay? Notice that sodium concentration is high outside of the cell, both on the tubule lumen side and on the interstitial fluid side, and it's low inside of the cell, okay? So our first step is pretty easy because we just need to follow our concentration gradient, right? We want to cross this apical membrane right here. We do that through a variety of mechanisms. So here we have a leak channel shown, but sodium can also move in through a variety of symport and antiport mechanisms as well. But in all of those cases, we're moving down our concentration gradient and we don't require an energy input. All right, now once we're inside of the cell, we need to cross this membrane right here. Since we're going to be moving against our concentration gradient, we need an energy input. So we use the sodium potassium ATPase to pump sodium out, okay? Here is a specific example of how we use our sodium gradients to transport other molecules, okay? So we're gonna use the example of glucose. And again, we have our tubule lumen here, our tubule wall here, and our extracellular fluid here. I want to point out that glucose concentration is high inside the cell and it's low outside of the cell, okay? So taking a look at our first step here, we need to move glucose from the tubule lumen into the cell. We're going to be doing that against its concentration gradient, okay? So the power from that comes from sodium moving down its concentration gradient, right? So we have symport happening here. Sodium is moving down its concentration gradient. The energy that is released from that is used to transport glucose against its concentration gradient, okay? Next step, we need to move glucose across this membrane right here. That's going to be fairly easy since we have a high glucose concentration inside of the cell and a low glucose concentration outside of the cell. So we're just going to use facilitated diffusion to move glucose across that membrane there, the basolateral membrane, okay? And then finally, just to kind of, for good measure, I guess, we're going to move sodium out of the cell using the sodium potassium ATPase. Okay, so I just want to mention this example was using glucose, but I want to mention that we have a variety of other molecules that are also transported in this manner using these sodium gradients to help transport the materials across the tubule wall.